Professor SNAB, where we aim to make A-level biology as simple as a cell. So today we're looking at a core practical in which we'll be preparing and staining a root tip squash to observe the stages of mitosis. So animal and plant cells both have stem cells. However, the range that the stem cells can specialize into reduces over time in animals. However, plants do not lose this range as they have meristems at the roots and shoots. Meristems are described as totipotent, which means they can differentiate into any cell in the plant. And the next, so we're going through a definition next. So it's the definition of the mitotic index. And this is so the mitotic index of a sample is the ratio of cells undergoing mitosis to the total number of cells in a sample. And it's quite important to know this because you will be asked, so there are different ways you could be kind of indirectly asked about this. And one way is like a 3 or 4 marker where you're directly asked to find the mitotic index of a sample. So yes, you just divide the number of cells going through mitosis over the total number of cells. However, you will see some practical questions, so questions on the practical, which are like 5 or 6 markers. And one of the bullet points is basically you mentioning finding the mitotic index. So if it's relevant to the question, then do mention this. So another point, so to know what cells are going through mitosis is you're going to look for the cells in which you can see the chromosomes. And this is because before mitosis, the chromosomes are condensed, which means you can see them under a microscope. So if you can see chromosomes, it means the cell's going through mitosis. So now we're looking at what we need for this practical. So you need the garlic root tip, 1 molar hydrochloric acid, toluidine blue stain, distilled water, watch glass, sample tube, pipettes, microscope slides and cover slips, forceps, filter paper, and scissors. So cut a 5 mm sample of the root tip using a scalpel and add root tips to sample tubes containing hydrochloric acid and leave this for 5 minutes. Set a watch glass containing cold distilled water and add the root tips to the watch glass for another 5 minutes. So after that, you dry the root tips on filter paper and spread the cells out using a needle and view the tip under a microscope slide. So the reason we do this is to make sure that the chromosomes are visible because that's your indication of whether a cell is going through mitosis. And then you add a drop of toluidine blue to the slide and let it stain for 2 minutes. And once again, that's so that you can actually see these cells under a microscope. After that, you lower a cover slip on the slide and ensure that air bubbles are not trapped between as you squash the slide so that your results are not affected. After that, you view under a microscope and adjust the objective lens from the lowest and then change the coarse adjustment knob and the fine adjustment knob as needed. So make sure your objective lens is going from the lowest resolution to the highest and not the other way around. So then you can actually find the correct point at which you can see the image clearly. Also, another thing is to ensure that the coarse adjustment knob does not clash into the slide. So when you're kind of adjusting it, just take a moment to look at the microscope from outside so that you're not breaking your slide. So mitotic index. So to calculate the mitotic index, you need to count the number of cells undergoing mitosis and the total number of cells. You need to then implement it into the following formula. So we've gone through this, but it's basically dividing the number of cells with visible chromosomes, because that's your indication of mitosis, over the total number of cells in the sample. So now we're looking at the risk assessment of this practical. So hydrochloric acid can cause irritation to eyes, or if you have cuts, hence it's important to wear eye protection and avoid contact with skin. And however, in case of contact, wash immediately with water. You will also be using toluidine blue stain, and it can cause harm or irritation when in contact with eyes or cuts once again. So just like hydrochloric acid, wear necessary eye protection and avoid contact with skin. However, in case of contact, wash immediately with lots of water. You will also be using the scalpel to cut the garlic roots, so be cautious while using it and use forceps to hold the sample when cutting. Apply pressure and elevate cuts in case of accidents and seek further medical assistance. And then finally, you have broken glass. There is the risk of cuts from sharp objects, so be cautious when handling anything that is made of glass and keep them away from the edge of desks. However, in case of an emergency, elevate cuts and apply pressure on the affected area. However, do not remove glass from the wound and seek further medical assistance as needed. So that is all from Professor Snap today. Hope you found this video useful. If you have any questions, let me know in the comments below and I'll get back to them as soon as possible. And don't forget to like, share and subscribe.